All right, greetings, mic check, 1, 2, 3. Good evening, or sorry, good afternoon. Can you all hear me?
Yep.
All right, so we are recording live from John Pennycamp in Key Largo. If you've never been here, you should come check it out. A lot of the rare and imperiled butterfly species are often seen here, but let's move on. I like to start with, okay, here we go. So let's say we have some butterfly experts here, and you think you might have the imperiled Florida dusky wing in your backyard, but is it really the Florida dusky wing? So we have option A, B, or C. What do you think? Which one is the imperiled Florida dusky wing? Is it flying around in your backyard? Now you can type in the chat, or you can just blurt it out.
B.
All right, so we have B. Do we have any other guesses, or C or B?
I go with B.
Well, Scott is correct. It is actually B. Mm-hmm. So that is the imperiled Florida dusky wing, and in Miami at least, there are people who actually sometimes see this in their backyard. So are you curious? Would you like to become a member? If you go to miamiblue.org, you can go to membership page, and it gives you the information on how you can become a NABA Miami Blue chapter. So please consider it. If you enjoy butterflies, gardening, you want to know more about them. And of course, we have our guest speakers who talk about all kinds of various topics and so forth. So welcome again. And now I'm going to turn it over to our president, Dennis Ali.
So good afternoon, folks. This is the one part live that the people who showed up in Pennycamp can hear live anyway. Welcome. This is our November meeting. We have quarterly meetings, and I'm glad you folks showed up. I apologize that you are encumbered as opposed to appearing remotely, but that's the problem with technology. It will get solved next time. Don't worry. So anyway, we've got a guest speaker who Jay will introduce, and then we'll go through a few items off of this agenda. I'll give a quick conservation report of what's going on, at least through our chapter. And Jason, I turn it back to you to introduce our speaker.
All right, so let me introduce Sarah Cabrera. Okay. All right, here we go. So our guest speaker today is Sarah Steele Cabrera. And in the beginning, she grew up in Rhode Island, where she discovered her love of nature, exploring oak forests. I'm assuming they're temperate deciduous forests, right? That's what we get up north. I'm from Ohio. And freshwater swamps. Currently, she is a PhD student at the McGuire Center for Lepidoptera and Biodiversity at the University of Florida. And she also attained her master's degree in entomology at the University of Florida. If you didn't know, the University of Florida, I believe, is in the top 10 regarding public universities in the United States. So UF is a very, very good school, and they're doing a lot of great research with butterflies. She has a lot of experience. She spent the past 6 years researching rare and endangered butterflies in the Florida Keys, but has also conducted biological field work in Costa Rica. I always have a hard time saying that state, but Massachusetts, St. Croix, New Mexico, and other regions of Florida. And so her presentation, which of course, we're the Miami Blue chapter, is going to be an update on the Miami Blue Butterfly Project and preliminary results from the Schaus Swallowtail Population Genetics Project. Yes, so thank you, Sarah, for being our guest speaker today, and we all look forward to your presentation. So I'm gonna stop sharing and give Sarah the floor. All right. You should have the ability to share now.
Yeah, thanks, Jason, for the lovely introduction, and thank you all for being here and for hosting me. Let me just get my presentation open here. One note is that unfortunately, I do not have preliminary results just yet. I guess I was being a little overly optimistic when I gave y'all my talk title. But I will talk about the project, just don't quite have results just yet, working through some of those analyses, but I will talk about the project. Can everyone see my screen? Sorry.
Yes, we can see your screen.
Okay, cool. All right, so like Jason said, my name is Steele Cabrera. I am a PhD student at the University of Florida, working at the McGuire Center for Lepidoptera and Biodiversity. I'm in the school, I'm getting a PhD in interdisciplinary ecology from the School of Natural Resources and the Environment, if you're curious. Okay, so to start out here, many of you probably heard quite a bit about the insect apocalypse, right? It got quite a bit of press starting a few years back, both in the scientific literature as well as in sort of popular media. There were several studies that came out reporting really enormous declines in insects in various places in the world. And then these reports were immediately followed by sort of a backlash almost of folks saying, no, there's not an insect apocalypse happening. And so I want to talk about this because a lot of our work in insect conservation is occurring with this as a backdrop, right? Are insects declining? Are they not? How are they declining? Where? I would argue that it's clear that insects, at least some of our insects, like our imperiled butterflies, are clearly in decline. But the jury is still out on whether all insects are declining in all places. But today I'm going to focus on a few of our species that we know are in decline. And butterflies in general, there's pretty robust data to show that most butterflies are indeed have been under consistent declines. This is a study from a colleague who actually worked analyzing NABA count data. I believe this is from Ohio, is where he did this study. And he analyzed over 20 years of data and found roughly 2%, a decline of roughly 2% per year, which maybe doesn't sound like a lot from year to year. You can see in this chart here, each of these dots is 1 year of data. But over 21 years, those are huge reductions in butterfly abundance numbers. And whether or not what these declines, the magnitude of these declines might look like in South Florida, we don't know. But likely something on this order or possibly greater, given what's going on in South Florida, the context. And so some common insect conservation strategies, of course, include habitat conservation and restoration. So a lot of what you see in Florida is, of course, re-implementation of fire. As many of you will know, most species in Florida are fire adapted. And so that's what this picture shows, is sort of the habitat pre-fire, immediately after a fire, and then post-fire, which provide completely different habitats. So that's incredibly important. And I put up this slide because I want to emphasize that at the end of the day, in order for any of these species to continue to exist, there needs to be good habitat existing for these species. But today I'm going to talk mainly about the last two bullet points here, captive propagation, as well as reintroductions and translocations. So what do you do if you can't, you can conserve and restore the habitat all you want, but if a species has disappeared from that habitat, you're forced to do some more extreme conservation methods, like what we've done with Miami blue butterfly and Schaus Swallowtail butterfly, so captive propagations and reintroductions. So the first butterfly I'm going to talk about today is the Miami blue butterfly, which a lot of you probably know about. So hopefully this isn't too much of a review for everybody. It's federally endangered and only found in South Florida. It's a little butterfly. If many of you are likely familiar with Cassius butterfly, it's about the same size as a Cassius, maybe slightly larger. And it was historically common. So I think it's a great sort of ambassador butterfly that you all have chosen for the chapter, because it's just really emblematic, I think, of some of what has happened with butterfly populations in Florida. And currently the only populations of this butterfly that are known to exist are in Key West and Great White Heron National Wildlife Refuges. So essentially the area or near and around Key West in the lower Florida Keys. So just as a bit of background for folks who maybe are newer to butterflies or just aren't familiar with telling all these tiny little blue butterflies apart, because it is not easy. There are 4, maybe 5 species of blue butterflies found in South Florida. The most common of which are the Cassius blue and the Serranus blue. And I get emails pretty frequently from folks who've taken photos of one or the other of these butterflies and then think they maybe have a Miami blue on their hands and want to report it, which is great. I love getting those emails because I'm always so impressed that people are looking at these tiny little butterflies and also are able to get good photos of them. For some of you who might also do photography of butterflies, you know exactly how difficult it is to get a good, decent photo of any of these tiny little butterflies that don't seem to like to land a whole lot. And so the best way to tell some of these apart is the Cassius blue here. You can see that it's got these stripes rather than really distinct spots like the Miami blue butterfly and the Serranus blue butterfly have. And then the main difference between the Serranus blue and the Miami blue is that the Miami blue has this white stripe here on its underside of its wings, whereas the Serranus blue generally doesn't have one or has one that's much less prominent. And then I put the Eastern pygmy blue butterfly in here because it is a member of the same subfamily and is considered a blue butterfly, even though they're a little bit more brownish in color. And they are found in salt marsh habitats, so usually found in different places from these other blue butterflies. They're also significantly smaller, and you can tell them apart because of these several black and iridescent spots rather than just 1 or 2. Also, fun fact, Eastern pygmy blue, while tied with possibly Western pygmy blue, are the smallest butterflies in the United States, which is pretty neat. They're pretty adorable. And I do put the Knickerbean blue up here. They are rather similar. However, these have not been recorded in Florida in several years. I've never seen one in my 6-ish years of working in the Keys. They haven't been reported in that time, but a way to tell them apart here is this orange spot is pretty elongated compared to the Miami blue, which has sort of a smaller, more rounded orange spot here on the underside. And an important note is that it is really important to get a good look at the underside of these butterflies to identify them from just when they've from above. If they've opened their wings, it's pretty difficult to tell some of them apart, and it's not necessarily possible to ID them to species from that upper side of their wings. All right, and just a little bit more review here. So the Miami blue has at least 3 larval host plants that we know of, 2 of which are Fabaceae, so in the pea family, Keys blackbead on the left, gray knickerbean in the middle, as well as balloon vine, which is not in the pea family. It's a little bit odd in that way on the right. And some other neat facts about butterflies. Some colleagues made this brief video, so hopefully the audio comes through. If not, it's also pretty neat.
The Miami blue butterfly is considered one of the rarest insects in North America. The caterpillar of this butterfly can be found in many different color forms, ranging from bright green to dark red. This caterpillar has a peculiar relationship with an unexpected organism, ants. The Miami blue caterpillars have evolved special organs to produce a sugary liquid from a small slit on their back called the dorsal nectary organ. The combination of this sugary liquid and dispersing chemical signals sent out by the caterpillars works to appease these otherwise vicious ants. These ants will drum their antennae on the backs of the caterpillars, which is a signal for the caterpillar to secrete this substance full of carbohydrates and amino acids. Butterfly larvae have a wide range of different predators, including ambush-style predators that hunt these caterpillars. Carpenter ants act as bodyguards for the caterpillars and will viciously attack any potential predators. While the ants are receiving a nutritious snack from these caterpillars, the caterpillars are receiving protection against insect predators. Just by having these ants around, the caterpillar's survival rate increases significantly. Native to Florida, the Miami blue can only be found in the Florida Keys today.
Anyway, some colleagues made that video about the relationship with Miami blue that Miami blues have with ants, and I just think it is a super neat video plus a really cool fact. So hold on, let me get out of this video. Does anybody have any questions so far about the history section here, the background? Okay, maybe we'll wait till the end to take questions, I guess then. Like I said, the Miami blue was once common throughout much of Florida. So this in red here is the historic range of this butterfly, really. So throughout the Keys, Miami, and then up along both coasts, pretty far north, like to the Tampa Bay area on the Gulf Coast, and up to Cape Canaveral on the Atlantic coast. And then down here, that's really been reduced to just a few islands within this yellow box, all the way at the bottom of the Keys. And really, why is this? It's all due to coastal, well, largely due to coastal development. On the left here is a picture of Miami Beach back in 1925, sort of right during that first big development boom in South Florida. And here is that same section of beach currently, well, 2018, so more or less currently. So you can just see the massive land use changes that have occurred, that there's just really not that much space left for this butterfly along much of our coasts. So back where this butterfly is currently extant, so where it currently exists, is largely within Key West National Wildlife Refuge. This is what these islands look like. They are beautiful. However, their elevation is maximum about 1.5 meters above sea level, so very vulnerable, of course, to sea level rise. And because it is a small area, these butterflies are also highly vulnerable to events such as hurricanes. And I will note that these are pretty large islands, but this butterfly is not found sort of throughout this whole habitat. It's really just found on smaller sections, sort of near the beach here in this habitat. Back here, most of this island is actually mangroves. This butterfly is not found in those mangroves. It needs a little bit more elevation for its host plants and nectar plants to occur. And so at the Florida Museum of Natural History, under the supervision of Jarrett Daniels, who is my advisor and my boss, have a captive breeding program for this butterfly. You can actually come, if you happen to be in Gainesville, you can actually see this breeding program in progress at the museum. So that window in the back of this is actually the window out to the public part of the museum. So if you come by, you might be able to see some of this conservation work happening. Here are my lab mates, Matt and Kristen, who are working with caterpillars that are in these vials, sort of sorting them and helping them to grow. So what we do is we actually cut pieces of the host plant for these butterflies so that we're not rearing them just on these giant plants because it's really easy to lose track of small caterpillars, and they also have a pretty high rate of predation by ants and other things. Not the carpenter ants like you saw in the video, but other ants will predate them. So there we rear them in the lab like this instead. And so now I'm going to talk a bit about an experiment I did in 2019 as a master's student. I spoke to the Miami Blue chapter, trying to think, it was about 2 years ago when I was actually in the middle of doing this experiment, or maybe right before I did it. So I'm here as sort of an update to that experiment now that I have results and I'm getting ready to publish them. So I'm going to go into the background for folks who weren't here a couple years ago when I last talked about this, or have quite reasonably forgot what I talked about since that was quite a long time ago. So the idea here was to do experimental butterfly reintroductions at both Bahia Honda State Park and Long Key State Park. These locations were chosen partially due to the habitat. There's abundant host plant available at both of these locations, and also because Bahia Honda State Park was one of the last locations these butterflies were known to occur. This population disappeared in about 2010, so it's sort of a logical point to restart is where they were last found. And what we did was we actually put up big field enclosures. So this is one of our technicians in the lab, Taylor Hunt, putting up these enclosures. And the reason we did that was actually just to be able to find these butterflies again. So we first did some trials of these reintroductions, and we let the butterflies go at the sites. But without these cages in place, we were just not able to re-site those butterflies later. They just quickly dispersed, and I was not able to find them again even the next day or 2 days later. So in order to be able to collect data, we found sort of an imperfect solution, which was to put up these cages over existing habitat. So inside of these cages, there is host plant, are nectar plants, and there's enough habitat in there for them to go through their entire life cycle. This is not really maybe an ideal situation, but it's really the only alternative we could figure out, because if we release butterflies and can't find them, it's really impossible to learn very much at all. So this was sort of our next best option. So my main question here for this project was, how does life stage at release affect the success of reintroduced butterflies? So we were interested mainly in pupae and adult butterflies. We tried larval releases, so releases of those caterpillars, and found that mortality was really high when we released caterpillars. So we were focusing mainly on pupae and butterflies for this, and to see, is one of these going to be better than the other as far as a reintroduction goes. So here you can see both an adult butterfly and a pupa. The adult butterfly on the left, when we released adults, we marked them. You can just mark adult butterflies with Sharpies. So it has this green dot here, which does not occur naturally. And this allowed us to differentiate butterflies that we reared in captivity and later released from butterflies that were born at the site. And then this is how we released pupae. We made these little PVC tubes that had big enough holes at the top for them to be able to crawl out as adult butterflies. And this is the size they actually are. They're very small little pupae. They actually, I liken them to like a piece of rat poop. They're about that size and shape and color. But if you take, say, like a macro photograph, they actually are quite beautiful. You can see like this is the developing eye and antenna and the abdomen and the wing here, but it's not always appreciable at our scale. So what we did was we released the butterflies into the cages and then subsequently counted them. There are some folks on this call now who helped count butterflies. So it was really due to the effort of volunteers that we were able to get all these butterfly counts done. So this butterfly counts allowed us to see how long are they surviving in these cages. And then afterwards, we would do egg counts. So if the butterflies are going to be successful in a reintroduction, well, they need to lay lots of eggs, right? And this is what those eggs look like at sort of a macro scale. They're rather beautiful and have cool patterns on them, but they are tiny. So this is my thumb, and I've circled in yellow 3 butterfly eggs. So super tiny. Taylor, our technician, and I spent many hours squinting in the sun, looking and counting eggs. So next, our results. First off, we had some encouraging signs that this work could work as a proof of concept. It did turn out fairly well. We had some short-term population reestablishment occur at Bahia Honda, which did not happen at Long Key. So at Bahia Honda State Park, this butterfly persisted for about 6 months after we stopped doing reintroductions for the season, which maybe doesn't sound like a lot, but it's important to note here that 6 months is actually about 6 or 7 generations of this butterfly. They have a very, very short life cycle and reproduce quickly and continuously. So they kept going for about 6 months, but this population did not make it through that dry season of 2019-2020. But we were encouraged that there was at least short-term population sort of showing that, yes, this can work. There's more work to be done to figure out exactly, what else we need to be doing, but encouraging nonetheless. So this is just a different way of looking at those same results, that this worked really well at Bahia Honda State Park, but did not work very well at Long Key State Park. We never found evidence of more than 1 generation post-release occurring at Long Key State Park, which led to more questions, of course. What happened? And why were these results at the 2 sites different? And the reason we think that the results were different between sites were actually due to different amounts of host plants at the 2 sites. So looking at all of the ecological factors that we measured, the one thing that came out as mattering quite a lot for these butterflies was the presence of knicker beans. So the butterflies did better, survived longer, and laid more eggs in areas that had more knicker bean than areas with the other host, blackbead. And as well, they did better in areas with lots of leaf litter on the ground. So that's in comparison to, say, bare ground, which in these beach systems is really just sand, which makes sense given what we know about this butterfly. They likely crawl down as late instar larvae. So when they're mature larvae, they're done eating, they crawl down their plant and pupate among that leaf litter, which probably gives them a few advantages. It probably allows them to hide a little bit better from predators, but also that leaf litter holds onto moisture for longer and stays cooler than, say, just bare sand, which as you might imagine, gets very hot and dry on a beach. So that's our best ideas of why they did better at Bahia Honda versus Long Key State Park. And then back to my original question, which are more successful, pupae or adult butterflies? And they actually did about the same, which was surprising. I had originally hypothesized that adult butterflies would survive longer and lay more eggs because, of course, there's additional mortality between pupae and adults. So some of the butterflies that we released as pupae, they didn't all necessarily hatch out or eclose, is the technical term, successfully to become adult butterflies. So it was a little surprising that the pupae performed about as well as the adult butterflies. It's also very convenient for us as a project because the pupae are much easier to release. On the left here, you can see this is sort of a modified pupal release chamber that came out as a result of these experiments. This actually gets hung in trees. So the larger PVC pipe here holds up to a couple dozen pupae. You can see they're starting to emerge there, and is capped. You can sort of see in the corner, capped to keep the pupae from blowing out, but it has big enough gaps for the butterflies to fly out. And it's actually hung in trees and shrubs with pretty light tackle fishing line, so monofilament line. And this really helps keep the ants out. We've done a lot of tests with ant predators, and they actually have a lot of trouble. If you hang it correctly, those ants have a lot of trouble sort of walking out and making it successfully into this PVC tube with the pupae in it. And so, of course, this was a small experiment. You can see here at the bottom, the extant wild range of the Miami blue butterfly, the little areas that we did reintroductions in 2019. But of course, our larger goal is to restore populations of this butterfly in other parts of its range, right? The butterfly is going to have a higher chance of not going extinct if we can get it in more places. So what we're working on currently is habitat evaluations and further population reintroductions. I think I saw Lydia on this call. She was a huge part of making this this part of our project a success. So she and other folks at Fairchild Botanic Garden, as well as Zoo Miami, worked together to evaluate several dozen sites really across the state of Florida. This is not even all of the sites. They came up with further sites, but this is a pretty good representation of most of the sites. So they evaluated many sites across that historic range. And now this winter, we are going back to sort of look at that work they did, continue evaluating, and hopefully be able to do future reintroductions, hopefully at a site in mainland Florida. We'll see. It does, of course, hinge on the continued success of the captive colony. So that sort of concludes the part of this talk on Miami blue butterfly. Jason, do you think I should take questions on Miami blue or talk about Schaus and then take questions on both at the end? I don't know which would be better.
Let's save it to the end. All
Okay.
right.
Yeah, so hold your questions on Miami blue for now, and we'll talk. This is a shorter part since I don't yet have results on Schaus Swallowtail. So this is a male Schaus Swallowtail in habitat, actually perched on its host, and you can tell the males and females apart because the males have these yellow tipped antennae. And this work, I always like to show this photo. You know, everybody always thinks that working with butterflies is literally butterflies and rainbows, and it is not always. I know some folks in this call have done quite a lot of work with Schaus Swallowtail and know how it is to do that monitoring in a South Florida summer in the hardwood hammock. So yes, you can see here, this butterfly has actually been marked. So the Daniels Lab and other folks from the Florida Museum of Natural History have actually been monitoring populations on Elliott Key within Biscayne National Park for several decades. And most recently that has been a mark release recapture project. I won't talk too much about it here, but it involves marking the butterflies with numbers and then later seeing if we recapture them, and that allows us to estimate population size. So a bit of background on Schaus Swallowtail. It was first listed on the endangered species list in 1976, later reclassified in 1984. An important note is, unlike Miami blue, which was historically common, the Schaus Swallowtail has never been a common butterfly, and it's also always had a small range of really just extreme southeastern Florida from Miami to Lower Matecumbe Key. And currently this butterfly is only present in Key Largo and Biscayne National Park. And we are also working to reintroduce this butterfly to Lignum vitae Key, which is down here with this red marker sort of off of Isla Morada. This butterfly also has a captive breeding program for it at the Florida Museum. This is actually the same countertop from the last picture that had Miami blues, but just in a different season, is now at this point when this picture was when this photo was taken is now covered in Schaus Swallowtail larvae instead of Miami blue. I do just want to recognize the huge efforts of my lab mates who make this work possible, because they are dedicated much of their time to just rearing endangered butterflies year round. It's 2 full-time people who do mostly butterfly rearing in the lab. Oh, and an important thing here, for folks who find a swallowtail caterpillar and are not sure which species it is, there are a few factors that help tell Schaus Swallowtail apart from, say, giant swallowtail, which is the most abundant, and also Bahamian swallowtail, which occurs in Key Largo. The best thing here are these osmeteria, is what they're called. They're sort of fake antennae. Really what they're doing here is it's thought that they are mimicking like the tongue of a snake, or otherwise just sort of scaring the predators. So if you were to gently poke the caterpillar, you'll see that Schaus Swallowtail has off-white osmeteria, whereas giants have orange. And also this butterfly has these purple spots, which giant swallowtail caterpillars do not have. All right, so some ongoing conservation work with this butterfly also involves reintroductions. And this began in spring 2020, which we were fortunate to be able to continue this work despite the pandemic. And these have gone on into this year. This is several hundred pupae and larvae that were released on the island. And so far, things seem to be going pretty well. Susan Coulterman and others actually saw adult butterflies on Lignum vitae Key in May of this year. And her and others, as well as state park folks, have been going back out there to monitor this reintroduced population. So it's early days yet, but we have some good early signs that that these reintroductions are having some success out there. Okay, so on to my population genetics project. So my hypothesis, or what I think happened, is that there were genetic bottlenecks which occurred due to a combination of urbanization in Miami and the Keys, as well as events such as hurricanes, namely Hurricane Andrew in 1992. This photo is from Elliott Key right after Hurricane Andrew. I know the photo quality is not great, but you can see there's not really a leaf left on any of these trees. And so for folks who maybe aren't as familiar with what a genetic bottleneck is, the idea here is that in the before an event, there is genetic diversity in a population. So each of these colored dots represents different genetic types. So that before an event, there's lots of diversity here, and then something happens. And it can be one event or multiple events. So, one event, an example of that would be like Hurricane Andrew, or it could be something more like more events over time, like urbanization. It doesn't all happen at once, but in the space of a couple decades, the land use patterns were very different. And so the idea here is that in a genetic bottleneck, you go from high genetic diversity to then something happens and only a few of those survive that event, represented here by just the blue and red dots. And then what happens is that just these individuals are left to mate, and then in the future, populations are much less diverse, as represented here. So there's less genetic diversity in the populations after a catastrophic event than there were before that event. And so my interest is to see, did something like this happen with Schaus Swallowtail? And if so, can we detect it? Sorry, I'm gonna go back to this. I put this out of order in this presentation. And so the way we're doing this is actually going back into the museum collection. So I am very fortunate to work at the McGuire Center for Lepidoptera and Biodiversity, which has over 10 million specimens of butterflies and moths, as well as quite a nice collection of Schaus Swallowtails. So what we did here was we looked at specimens, some of which were over 100 years old, and extracted DNA from those specimens. Now the ones from I think the oldest specimens we looked at were from 1903. We weren't able to successfully extract DNA from specimens quite that old, but we were able to successfully extract DNA from specimens like these collected in 1945 in the Keys. And so the idea here is we're going to compare the DNA from these old specimens. So from the I believe we were able to get some from the late 30s and in the mid 40s, and compare those to specimens that were taken in the 1970s, as well as those both pre and post Hurricane Andrew, and see if we can determine, was there more genetic diversity in the past than there is today? And in addition to looking for the population bottlenecks, we're also hoping to learn about the population structure, current population structure of this butterfly. So this is the same figure I showed a couple slides ago. This current range of this butterfly is, Biscayne National Park down to Key Largo, as well as our reintroduced populations here. The question is, in this current range, are butterflies intermingling? Are they mating between all points across this range? Or are, say, butterflies from the northern range different from those found in the southern part of Key Largo? I would hypothesize that because this is a large, strong flying butterfly, that they are likely making these trips across the range fairly frequently. But it's important for us to know that going forward, since we're putting them in this reintroduced population here, well, how far can these butterflies fly? That's really difficult to actually do a study, like in the field. That's pretty difficult because, well, you'd have to mark and catch recatch quite a lot of butterflies to be able to figure that out. And they also, they fly over water. We're not gonna be able to chase down these butterflies. It's likely that most of these butterflies stay rather close to where they hatch out, but maybe every once in a while one makes that flight of a couple dozen miles between say Elliott Key and Key Largo. And so this will help us to inform further conservation and management of this butterfly. So having this sort of radius, we'll be able to look at how far apart can we put a reintroduction site to maintain connectivity with that extant range. And so the way we are doing that is by collecting larvae and eggs across the range. This is immature butterflies. And I would like to note that this is something we're doing anyway. We're not sort of killing butterflies for no reason. We are already collecting these eggs and larvae for the captive colony. And so it's not just that we're taking them, they are being incorporated into the captive colony and bred out, and we are taking that genetic sample only after the natural death or using tissue that's non-destructive. So we were actually able to successfully extract DNA from the shed skin of both the larvae and the pupae, which was pretty neat. So you can actually get DNA from these animals without killing the animal, which was pretty impressive for such a small organism. And this gives us a couple things. We are hoping to increase the genetic diversity of the captive colony, but also trying to figure out what's going on with these populations of butterflies in South Florida. So yeah, I don't have those preliminary results just yet. So I'm hopeful to have them in about 6 months' time, that I should be able to analyze some of those DNA sequences. We sent them off for sequencing, got the sequences. Now I need to figure out how to analyze them. So lastly, I just want to give a huge thank you to my lab. This is, some of you will know Jarrett Daniels, the principal investigator of these projects, as well as my lab mates who make this possible, as well as many, many volunteers who have worked on this project over the years. Yeah, it really would not be possible without awesome volunteers and folks helping. And if you want to get in touch, this is my email here. You're welcome to email me with any questions. We have a lab website here, and both myself and the lab are on Twitter. So yeah, with that, I will take questions.
Well, thank you, Sarah, for presenting and talking about the blue and the Schaus. So let's open the floor. Let's go. I'm pretty sure there are plenty of questions, comments, concerns. So go ahead.
I have a question.
Yeah, go for it.
You were mentioning that the population is, or you're trying to save it from becoming extinct, but you mean extinct in the US, right? Because we have populations in the Caribbean of both Schaus and blue, right?
Yes, that's a good question, and really important that you brought that up. So both of these butterflies are related to butterflies in both the Caribbean and the Bahamas. However, sorry, I have some baby chickens. If you hear chirping, I didn't really have another place in my house to do this today. Oh, no. So there's some little baby chickens in a corner of my office right now that are being really loud. So sorry, they are related to, but not the same as Miami blue or Schaus swallowtail that are located in the Caribbean. There's been some genetic as well as, so studies looking at both the genetics as well as the morphology, or how those butterflies look between the Bahamas and the Caribbean and South Florida. And they are actually sufficiently different. It's actually been proposed that for both Miami blue and Schaus, that they are sufficiently different that they should actually be considered a separate species rather than subspecies of the same species. I am not a taxonomist, so I will leave that to the taxonomists to decide. But however, there are significant differences between these butterflies in South Florida and the related butterflies in the Caribbean and Bahamas.
And I was going to ask you about that, the taxonomy, because in iNaturalist, it popped up that now Schaus has its own thing. It's now, I think, Heraclides pontianus instead of Aristodemus pontianus.
Yes.
So it out there is already renamed as its own species. I don't know if it's true, but
yes, that is true. And I guess, how it's interesting that iNaturalist, it's up to the curators of those particular groups of taxa on iNaturalist. There is good evidence for it. If you're interested, I'm happy to send you a PDF of the paper that they based that off of. They didn't just, the folks on iNaturalist didn't just decide on a whim to rename it its own species. That was based on an academic paper that I think was published maybe 2019 or 2020, I can't remember now, but if you'd like, I'd be more than happy to get you that PDF. I'll also just put my email in the chat again, so that folks have it.
Now, definition of a species, isn't it just to breed them to see if they have offspring? Can you get Schaus from Cuba or Hispaniola and breed them with Schaus here and see what happens?
Yeah, that's a really good question that I am not going to be able to satisfactorily answer. So species.
Just giving you ideas for your peers to do more species.
We are actually doing some of this work, so we're trying not to repeat the work that those folks did that made that determination about that Schaus is a separate species. But we did actually extract DNA from Schaus specimens from elsewhere so from Cuba, from the Bahamas, from Hispaniola. So we are working on looking at some of that just because we're looking at different locations on the genome than they did in that study. But anyway, sorry, back to species concept. So it depends who you ask, and it's a rather controversial topic. So some people would say, yes, a species is, can those individuals successfully interbreed? Whereas others would say a species is individuals that can and do successfully interbreed. So they could interbreed, but do they actually have the chance to? So that's usually separated often geographically. So, do butterflies, say from Cuba, often interbreed with those from Florida? Possibly, but the genetic data we have available say that that probably doesn't happen frequently enough, and that because of that geographic distance, it's likely that they are different enough to be considered separate species. I hope that makes sense. And like I said, I'm not a taxonomist, so I'm going to let them duke it out on this one probably. It does bring up a good point, though. You know, what at the end of the day, what is the utility of calling it a subspecies versus a species? And I would say some of this, you know, comes down to sort of what one values. We can list subspecies on the endangered species list. So at the end of the day, does it matter whether they're subspecies or their own separate species? I'm not sure. Maybe, maybe not.
Well, it matters if you're trying to save a species from becoming extinct.
Yes, that's true. Yes, that does. I guess I would argue that yes, that does matter, right? If it's extirpated, just, you know, not present in an area it used to be versus extinct, doesn't exist anymore.
So here, let's let Ricardo come in. He's raised his hand, so I'm pretty sure he has. All right, Ricardo.
Hey, thanks. Good job, Sarah. So for the Miami blue butterfly reintroductions, how did you measure success with pupae in the adults?
Yeah. So with those pupae, we were able to, because of those little PVC tubes we had them in, we were actually able to go back in a few days after we released them, and we're able to see whether butterflies successfully closed or came out of those pupal cases, or were still in there that died as pupae, or that were predated by something. You can actually, that's one of the neat parts about releasing pupae, you can actually sort of see what happened. And with the adults, it's pretty hard to actually find the dead. Once they die out in the field, they, you know, pretty much disintegrate or disappear. So what we did was we looked at the success rate of those pupae based on our visual examination of those pupal cases. And then the adult survivorship is all based on those counts. So we know how many butterflies we put into the cage, and then know how many butterflies we saw in subsequent days. That makes sense.
So if you saw them, I don't know, 5 days later, that would be considered success, or 10. I don't know.
Yeah, so what we considered success for an adult butterfly is surviving to at least 2 days post-release, because based on what we know about the biology of these butterflies, you know, their lifespan as adults is really, really short, like maybe a week if they're lucky. And so based on what we know about this butterfly, we put 2 days as success, because in those 2 days, that's enough time for them to sort of dry their wings, mate, and lay eggs.
Okay.
Good question. That's a really good question. Thanks, Ricardo.
All right, thank you.
Sarah, I have a quick question, and then Dennis has a question too. What can we do as butterfly advocates? What can we do in schools and with our neighbors? Do you have any suggestions?
Yeah, thanks, Jason. That's a really good good question that I did not get to in this presentation, and should have put more about that in there. I get that question a lot, specifically for Miami blue and Schaus Swallowtail. You know, if you happen to live somewhere that's very close by to their wild ranges, you know, planting their host plants and nectar plants is great, but I like to instead focus more on sort of the larger goals of butterfly and insect conservation, and really biodiversity conservation in general. So that, you know, is planting, if you have a yard, you know, planting it in native plants wherever possible. But more importantly, I think, is just, you know, not using chemicals wherever possible on your landscape, because there's life everywhere. You know, even if you live in a very urban area, there's biodiversity right there. So just trying not to use pesticides where possible in your landscape and in your house is really important. Planting native plants where possible, and of course, I think the biggest thing is educating others. You know, folks can't value the biodiversity that they don't know exists. So I think, making folks aware that the biodiversity we have here in Florida is special, and that there's stuff that's in Florida that is in danger of disappearing, and it's worth conserving is really important. So I would think those are. And the other one is turning off for many insects, turning off outdoor lights that you don't need at night. Certainly we need outdoor lights to, stay safe, but if there's lights that aren't in use, just turning those off where possible.
All right, thank you. And Dennis, go ahead.
Sorry, there's a lot of back noise, but Sarah, can you put the slide back up that showed the historic range of the Miami blue?
Yeah, give me 1 second here. Scrolling through. Ooh.
I'll just note that lights out is good for migratory birds.
Yes, thank you, Barbara. Yes, it's important
and
for
bats.
Yes,
Yes,
important
so-
for more than just insects.
Sarah, and it may be that the map is more stylistic than accurate, but it strikes me that there is a lot of undeveloped habitat sitting in the southwest portion of that arc. And I know I've talked to Jared about this 10 years ago, about what's going on in Cape Sable, essentially the 10,000 islands, all of that. How confident are you that that's the historic range of the butterfly?
So let me make sure I'm understanding that. Well, I would agree first off that yes, this map is a bit stylized. The idea here is sort of showing roughly the northern extent, and showing that this is generally coastal. Was it in every single spot? We just don't know. There's not a lot of specimens, because I think this butterfly just wasn't really of that much interest until it started disappearing. I have an old copy of, I think it's a Peterson's Guide to Butterflies from 1945, and Miami blue is just listed as common with no other remarks. So I will say that, I guess, is that this is not, it's hard to be entirely accurate here with that historical range, but just to make sure I'm understanding your question correctly, is your question whether or not we can say that this butterfly occurred in Southwest Florida?
Yeah, I mean, obviously I am a believer that it's habitat destruction, but the naysayers look at that map and the obvious conclusion is, wait a minute, starting at the southern tip of the peninsula of Florida, Gully, all the way up through the 10,000 islands, there's no habitat destruction there.
Yeah.
So it's just, and it may be that there's just not enough records. I'm not familiar, but I've not done a search. Again, I suspect it was much more coastal than what that map indicates. Almost like what the Schaus is, very, very coastal related, and maybe just pockets up and down those coasts. But that's just my own observation.
Yeah, no, I think those are really good observations. Yeah, I do think this map likely goes too far inland, and is probably not a perfect representation. So yes, to sort of respond to that, what is this pressure? Largely, I would say it's urbanization, but I think the other part of this, why would a butterfly disappear from habitats where there is not that urbanization going on, such as maybe Cape Sable or 10,000 Islands? I think a lot of it has to do with sort of the way this butterfly functions in what we call a meta population. So where there's spots in this map where this butterfly would be abundant, and if they're able to disperse out from that population center, they'd likely be found in more places. But if you say, develop Miami, where it was once where it was thought to be the most abundant, is Miami, hence its name. You really limit that butterfly's ability to disperse into adjacent natural habitats. That's sort of one of the thoughts with this butterfly is, yes, while there is definitely habitat available for this butterfly still, a lot of these areas that are undeveloped are far apart from each other, right? So there's down here in these islands, that's quite a considerable distance from some of these islands to, say, Cape Sable, or some of these other spots along the coast, and that over time you can lose populations due to urbanization in other areas, if that makes sense. And like you also alluded to, at the end of the day, we won't know exactly how these declines occurred, because nobody was collecting, this type of population-wide data at the time that they occurred, that the declines occurred back in the 70s and 80s.
Yeah, thank you.
Well, thank you, Sarah, for your presentation and knowledge. And so, obviously, if you have more questions, she has an email address, so continue the conversation. Now we're gonna move on to the second part. Where are we? All right. Oh, wrong way. Okay. All right, so we did our Q&A with Sarah Cabrera. And now, Lydia, can you update us on the Key Biscayne project? Can you just briefly talk about what's going on? And this actually has relevance to the Miami Blue project. So I'll just let Lydia briefly talk about it.
Hey everyone, great presentation, Sarah. It was awesome to see all the different updates, especially about the reintroduction project. So Jason, myself, and Sandy Coy, we were introduced to Jorge Diaz Cueto, who is a businessman on Key Biscayne. Barbara McAdam actually introduced us all. So we've just been working, putting our brains together on some coastal, Key Biscayne coastal butterfly plants and target butterflies as well. So we developed for the project a target butterfly list. So imperiled butterflies and their host plants. Our idea with this is just to supply this information to Jorge, who can get the word out to his different connections, different residents on Key Biscayne, different businesses, possibly condominiums, the village of Key Biscayne proper, some of their buildings, just to get them familiar with some imperiled butterflies that they can help, and using this project plant list, which is different shrubs and different coastal plants that will do well in their coastal yards, and also information where they can find these plants, source the plants from nurseries. So we're compiling this information and providing it to Jorge. Do you want to add anything, Jason?
No, that was good. So this is just be on the lookout. We will continue to update you every quarterly meeting. All right, thank you, Lydia. Next up, we have the conservation report by Dennis.
Yeah, thank you. Just I'll face you guys. Just three things on conservation. One, the Miami tiger beetle, which is, believe it or not, relevant for us, exists in Zoo Miami. It overlaps in some significant respects with the critical habitat for the Schaus Swallowtail. I'm sorry, let me try that again. Bartram scrub hairstreak and Florida leafwing. So the more critical habitat that's designated, the better it is for all of the species in that area. So we've joined in a letter, two letters. One that asks for a public hearing on the proposed critical habitat, and Adam, I'll send you a copy of the map of what's proposed. if I haven't sent it to you already. And also I'll send you a letter, which I expect a copy of, where we've asked, there was a comment letter provided by the Center for Biological Diversity on the importance of protecting the beetle. Obviously, it's sort of I'll call it an umbrella species. It's just another species in that area that needs protection. And the more that what's happening is that area around the Richmond Pineland, around Zoo Miami, has become a very important hotspot for endemic species. So leafwing, hairstreak, Miami tiger beetle all share that sort of area. And the tiger beetle is next, has been already listed. The critical habitat is about to be designated. So we're making a big push about that. On a related topic, Miami Wilds, which is the water park that's being proposed to be built in one of the parking lots of Zoo Miami, is something that we are taking a opposition to. The county hasn't signed the lease for the developer yet, but that's imminent. We'll see what happens. The two leading environmental groups are Bat Conservation International, because there's a rare bat there, the Florida bonneted bat, and tropical audubon, and sort of wearing a couple of different hats, but it's an area, again, because of the bat flies all over there. Its critical habitat is also being designated. Total overlap, or significant overlap, with the leafwing and the hairstreak. So it's just making the point that if we protect the area for any one of those species, it helps all the others that are there. So you've got, besides the two butterflies that have critical habitat in the area, there are two more that are teed up to overlap there, the Miami tiger beetle and the Florida bonneted bat. So we're, I'll say, active in the sense of joining in letters and encouraging the U.S. Fish and Wildlife Service to designate, not just designate the habitat, but designate appropriate habitat. More into suburbia, that's true. City of Coral Gables has reached out to several of us to ask for help in terms of pollinator plants and a list of native plants that they could use in their plantings in the city, in their parks and swale areas, et cetera. So we're working on that. Barbara has been helpful there. I'm working with her. But the city is really engaged in the aspect of pollinators and trying to do some type of restoration that would encourage pollinators in the city. And I've been spending a lot of time, I'd say one day every week, talking with the city of Coral Gables about that. We're talking email, something like that. So if anyone's interested in getting involved in that, I would welcome help in that regard. In a way, it's positive because we've actually got the government wanting to do the right thing. So the city's got lots of parks, pocket parks around, and they're anxious to put the right plants in that would attract pollinators. So again, our reach is beyond butterflies, but obviously what's good for pollinators generally is probably good for butterflies specifically. So it's an area that I want to encourage the city to do the right thing, and they're motivated. So that's an important entree. And I think if we can show that it works in the city, it could be, my selling point for the city is you can be distinctive, you can show off, have something to brag about, and maybe it'll create some pressure on everyone else, including Miami-Dade Parks, which is doing it as well. Barbara McAdam has been a significant player in that regard. So I think the county gets it, but the county's a far-flung operation. There are parks, there's EEL, there's public works, there's lots of different public lands that are under various jurisdictions, and just getting the county to be motivated to do the right thing in all aspects is an important aspect that it requires almost daily vigilance and watching out for what they're doing. And the final thing is something I'm tinkering with, but I haven't gotten traction yet, is, and it was mentioned in our program, I am overwhelmed by how much insect spraying is done in private yards in my neighborhood. I'd say over half of my neighbors are spraying for bugs. You see the trucks. We're against bugs. We want to destroy everything, yet on the side of the car, they have a monarch butterfly. So we destroy bugs and there's a monarch nectarine, okay? It's like unbelievable. I don't know what's going on there. I know it's somewhat regulated, but it's regulated at the state level. I know that the city of Coral Gables has stopped spraying. all of their properties. My big concern is just based on my walk around, when I'm walking the dog in the neighborhood and seeing the little signs, I'd say half of my neighbors are spraying their yards. And that's I assume, kills everything. It can't be specific to mosquitoes. And we need to figure out what we can do about that. I don't know if it's supposedly regulated by the state. The state doesn't have a clue about what that guy's doing, walking down in my neighbor's yard, okay? He's wearing a respirator of some sort for probably good reason. And they put the sign out that says, don't walk on the grass for 24 hours. But my concern is that stuff, besides we all should be worried. I don't know what it does to our water table, but it certainly is destroying every pollinator that's in the neighborhood. So I'd love to get some science on that. I don't even know what the chemical that's used is. I don't know who regulates it. So this is beginning at the beginning, but it's an area that we need to focus on. And obviously there's going to be a huge pushback both by neighbors and by the industry, because everyone who has chemicals wants to use chemicals because they like to sell chemicals. So I don't want to be negative about it, but it's a big problem. And I'm not sure what to do, but I'm going to sort of devote some time on that. Anyone that knows anything, you got my email, or you want to help, any help would be welcome. That's the conservation report. Yes, yeah. I'll repeat the question.
So Dennis, real quick.
Hang on. Yes. Yeah, it's the Florida bonneted bat. Yeah, so the question for the people who didn't hear that question from the audience here at Pity Camp about the Calusa Golf Course, which is under redevelopment. It's maybe a euphemistic word. This is what little I know. Tropical Audubon has taken a role in that, along with some of the residents, to try to protect that area and dissuade the developer from going forward. Don't know how successful that's going to be. There is a rookery in the island there, and it's it's God bless nature. If you leave it alone, it's a derelict golf course, and guess what's happened? It's rewilding. Okay, and I don't know anything about pollinators there or butterflies. We can talk separately about that. Yeah, the Florida bonneted bat is in that area as well, as far as I know. But I'll talk with you separately about that. I think there's a hearing November 17th. Okay, we'll chat. Barbara had something to say, and I will now shut up.
Yes, I just put into chat that anyone who applies pesticides or insecticides have to be licensed and has to pass a state exam. We do those trainings modules at the extension office, and it's been ongoing regardless of COVID. If you it might be a domino effect. If one neighbor sees their neighbor getting everything sprayed, maybe they think it's just a good idea because all bugs are bad and they're not aware of collateral damage. So you can do a little grassroots, talk with the neighbors, but let me know who you're seeing on those signs. I'll just, sort of surreptitiously check it out.
I just want to briefly highlight something. I'll keep it really, really quick. The neighbor-to-neighbor thing is not, we have to do something bigger than that. It can be something as simple as putting billboards on the highway, talking about pesticides and pollinators, to having a McDonald's map. Like when you have a tray at McDonald's, you could use that as information about butterflies and pollinators and stuff. But I mean, just kind of having a conversation or trying to come up with regulations. There are so many education opportunities out there, collaborations with schools. So I'm assuming in our next officers meeting, we should definitely put this as a big topic how can we reach people in a less aggressive way, right? But then maybe coordinate with businesses so that they can be partners in that. That's all I have to say
Right.
about that.
Because right there with loss of habitat is spraying with chemicals. So it's huge.
Now, Barbara, you and I can talk separately about that, but
Right,
sorry, I
got it.
asked you about it because you're more knowledgeable than I am.
Yeah, but if you're seeing them pop up in certain, spots, and you see neighbor after neighbor, then maybe the conversation would work as well.
Yeah. Okay, we'll continue that. That's my conservation report.
So do we, Linda, you want to talk about the nominations? Linda, your mic is on, but we don't hear you. Are you talking?
Can you hear me, Jason?
Yes, we can hear you now.
For some reason, I lost the screen. The slate of officers is the same as it was for last time. The only change is the position for secretary and the, I'm sorry, membership, and that position has been accepted by Mary Jackson. And all of the officers are listed on the Miami Blue webpage. And there will be an election at our February meeting. Can you hear me?
Yes.
Okay.
All right, thank you, Linda Evans.
I lost the screen for some reason.
Well, we can still hear you, so at least you're here with us.
Okay, okay, I just, on the chat, I just posted the slate of officers.
Okay. Do you want me to stop sharing so you can share your screen?
It's not, it's on the chat.
Okay. All right, so everybody just direct your attention to the chats, which I have. You can, well, for us in person, you can see it on the left side of the screen.
And then also it's on the webpage. The slate is essentially the same. The only change is we'd like to thank Patty Ferries for the number of years that she has worked so hard for our chapter, and she is asked to be, she'd like to step down as membership chair, and Mary Jackson has agreed to take over next year.
Okay.
So that's it.
All right. Next up, we have other business. So this is where we open it up to the floor. What's going on? What's happening in South Florida? Any other business? Anybody want to talk about those new Schaus numbers that was reported? The Schaus numbers that were reported? I know we have people from the Florida Keys. You were a part of the Schaus surveys in Key Largo. So what happened?
As far as I know, we had more than 800 Schaus in Key Largo this year. And then on Elliott Key, there were something like 1,300. So the numbers, the total number, I think, is 1,753, which is incredible considering in 2010 we had 0 in Key Largo. Just absolutely incredible. And it's the thanks to the team. There's more than 15 to 20 people that do surveys April, May, and June in Key Largo. And it's thanks to them, particularly Susan Colterman and Gail and Dale Kalasa and Mary Rose and all the other members, Miami Blue members that schlepped down to Key Largo to endure the mosquitoes and the heat and everything else just to present those numbers for us. So very, very encouraging, very, very excited about the numbers.
All right, thank you. do we have any other business before we adjourn?
No, we're just on a roll installing pollinator gardens into parks, both with Miami-Dade County, Parks, Recreation, and Open Spaces, and the new president of Coral Gables Garden Club. So that's been really good and really busy.
All right. So if we don't have anything else, thank you all for attending our quarterly meeting in November. We actually had a sizable audience that attended in person. I'm counting right now. I think we have 2, 2, 7. We had about 10 people that showed up in person. And so our next quarterly meeting will be in February, the first Sunday in February, and it might be at Castello Hammock, or that can be TBA. And we'll probably do the hybrid format where we'll have it through Zoom and also in person. So remember, if you want to become a member, visit miamiblue.org. If you're already a member, thank you. And enjoy the rest of your Sunday.
Thank you, Jason, for the technical aspect.
I try.
You got us there.
All right. See you later.